the cloud. Yes, so TJ today will tell us about one of the major achievements of last year, arguably.
Yeah, no, thanks, Sergey. So today I'm going to be going over AlphaFold. Specifically, this is the second paper that they came out with concerning the AlphaFold 2 algorithm that happened from 2018 to 2021. Today, sort of the overview that I want to go through, and we will get to how the structures can iteratively update in this form, but we're going to go over what is the folding protein problem anyway, what was so novel about AlphaFold 1, what did they change about AlphaFold 2, any kind of future work, and then we can answer the question, did DeepMind actually solve proteins? And so, especially for me, I had to do a little bit of background on sort of what is the actual problem that they wanted to solve. And so just as a little bit of sort of depth on this field, proteins carry out many of the important functions in the body and in biology in general, digesting food, contracting muscles, firing neurons, powering the immune system. David Baker, who's a very famous computational biologist and bioinformatician over in UW, has said that almost everything in biology occurs because of proteins. And so thinking about a general scale, they are around 3 to 6 to around 10 nanometers in diameter, which significantly varies from protein to protein with both molecular scaling and atomic scaling. And so when thinking about the protein firing or the protein folding problem, it really began in '72 with the Nobel laureate Christian and Fernandez in chemistry, where the protein's amino acid sequence, so the letters that we typically get, should fully determine its 3D structure. So this was the postulate that this Nobel laureate came out with. And so for nearly 50 years, we've been trying to predict sort of this 3D structure from amino acid sequencing. We got Leventhal, who postulated that it would take longer than the age of the actual universe to brute force many of these complex 3D structures of proteins, which then led to his paradox, which stated that protein structures fold spontaneously in nature and almost instantaneous, causing this conflict of why proteins can fold so instantaneous and rapidly, but also maintain structure in functionality. So it led to this notion that nature must be using a predictable folding pattern to arrive at these native states of proteins to be so reliable and rapid. So there was some conservation with evolution that is occurring. So previous computational methods were not practical enough to be clinically relevant. They started this challenge called the CASP challenge, and I'm blanking on, I didn't write it down, but I've been blanking on what the CASP stands for, the acronym, but it's every other year, top researchers from around the world will submit to the protein data bank, or the Worldwide Protein Data Bank, and use all known biologically relevant protein structures and the metadata associated with this. Just for any kind of clarification, X-ray crystallography costs around 12 grand per single protein run, and you need specialized scientists that almost require full dissertations amount of work for one single protein to be happening. DeepMind got involved in CAFS 13 in 2018 and then CAFS 14 in 2020. Each of these were the respective AlphaFold algorithms. So AlphaFold 1 happened in 2018, AlphaFold 2 happened in 2020. And so this is where they showed off and that they have a good benchmark in progressing sort of the field in a very robust way. I want to pause there. Does anybody have any questions about sort of the problem itself right now before we jump into more algorithmic stuff?
Can you elaborate on Anvinson's statement, the first one? How did he come up with that?
So I'm not very good at chemistry. I honestly have not taken chemistry since 9th grade, but from what I know is, given the amino acid sequence, which is just a series of letters, they want to be able to take that and somehow predict the 3D structure of that protein. And so that has been an open question for the last 50 years. Does that make sense? So the way that the atoms and molecules are interacting will actually change the 3D structure based on every single individual atom and molecule associated with the protein. Does that make
I
sense?
understand that. I understand. I'm just focused on this word. He said should, So the sequence should determine structure. So what is the basis for saying that it should?
Yeah, we could potentially go into further details, but I don't know if we should. It's more of just a postulate. I don't know what the surveying evidence that got him the Nobel Prize for this work that made him justify that statement, but we can take that as an assumption.
Is it a true statement? Do we know?
Yes, it is a true statement based off of evolutionary conservatives, based off of MSA stuff that I'll get into actually in the next slide, based off of historical evidence. We should be able to fully determine the 3D structure based off the amino acid sequence.
I think one reason maybe that he said that is because proteins fold similarly in different fluids, like with different other molecules available. So then it must be because of something else. Like it can't just be because of the environment, because I think proteins fold similarly in different environments. And I think that's why maybe it's also should, because it could be that there's partly some interaction with but I think generally the point is it doesn't need anything else. Like it's environment independent, which means that it must be intrinsic, the knowledge about the way it folds.
Yes, and actually going further on that, we can actually talk about multiple sequence alignment, or MSA, which is a huge part of this algorithm, where they made the observation that when studying different evolutionary lines of an organism, say like different lines of rats or different lines of nematodes up to different amphibians, they noticed that the 3D structure of proteins are much more stable over time than the genetic sequence that encodes the protein. So that means that the structure is, or mutations within the genetic sequence are quite common, but nature changes the structure much rarer. So when thinking about a tail needs to still function, a tongue needs to still taste, gut needs to still take in sort of these acids to break down foods. Those need to be conserved over an evolutionary iteration. So structure is rarely changed, but genetic information is quite common. So thus that leads to the postulate that proteins need to keep carrying out the same function over the course of evolution. So that means that the sequence is then conserving structure rather than genetics. So that means that using evolutionary data, the sequence can be run through databases of known structures from evolutionary lines to predict that proteins might be correct for the structure. Does that make sense? Does anybody have any questions about that?
I have a question about why is the 3D structure important for us to know? 3D, right?
It's because proteins can interact with other protein structures for complex interactions. The way that they are structured has sort of like a key and lock pairing, for the best analogy that I can get without getting into chemistry. The key and lock effect will actually change the structure of the protein, but actually perform different mechanisms and operations in like protein machines. Does that make sense?
Yes.
I think one nice their podcast, DeepMind's podcast, they also talk about this. So what they use this for example, is they use it to predict the 3D structure of proteins that seem like they could be helpful in solving diseases, for example, because otherwise it will take like 4 or 5 years to know what the 3D structure is, and only then you will know whether it can actually help a disease. And it's also for they're also using it for breaking down plastics in the ocean, because there are certain proteins that can break down plastics back to its original components, and some of these proteins that can probably do this, we just don't know their 3D structure, so we can't figure out if they actually do that. But if you can predict this within 10 minutes, then that's obviously a bit better than having to do it over 4 or 5 years, at least for like a first result. So that's why this is like really helpful.
Yeah. And so this kind of drives the question, why not just use the sequence data to just black box to the 3D protein structure? And the big reason is there's such a vast amount of array of data about evolutionary lines within the PDB or protein data bank, that it is, you should use as much information as possible. And so they're leveraging all the current information that is out there within the bank to better inform these models to get better accuracy. And so from here, can you guys see my cursor?
Yeah.
Awesome. So here we have something called a distogram. This is the 2D distance array between every pair of residues, which is sort of the different parts of the protein. So at least when I was reading this and without a background in chemistry, residues to me, and if someone has a better explanation, are just the different components of the protein itself. So it could be a branch, it could be a very important part. It could also be the different physical interactions of the atoms itself. But this residual or residue is then taken into distances between each residue of the protein. And so here in the middle, we have, this is an example of a distogram where it is a symmetric matrix which is predicted by sort of 2D projections into create a 3D folding pattern. And so as you can see here to the right, these are different distograms that have different weights to them that can be concatenated to then project to a 3D folding structure. Great. And so the question is, why not just use 3D Cartesian coordinates? Well, the distograms here are invariant to rotation and translations, which makes the target much easier to learn. And so that was definitely a design choice by the DeepMind team.
Are they unique?
They should be. I believe the complexity is unique. I think there are, because it's mostly the there's both the distance between the residues and the structure itself based off of the backbone template. And so there's like 7 different moving parts to this algorithm. So I'm sorry if it sort of is a little convoluted right now. But from my understanding is these residues are going to be updated based off of different angles and backbone structures as it iteratively updates.
Yeah, but I'm thinking one gram matrix, distogram, can correspond to multiple 3D folds. Like you can construct different 3D arrangements that will have the same gram matrix distogram. Yeah.
Yes, which is actually why they do predictive modeling here. So rather than predict just the target distogram, they actually do a distribution of 64 different distance spins between 2 residues. So they try to concatenate the predictions at 64 different possibilities between each residue.
Great, thanks.
Yeah, of course. And so finally, they'll use gradient descent to predict the phi and psi angles of the atomic structure. So not necessarily knowing the physics behind this, I imagine these are very specific angles that atoms interact with together. And so they use a scoring function that combines these predictions of the neural networks to the physical simulated forces. So here, whatever the phi and psi interactions with the strong and weak gravitational forces that can be interacting between the atoms. And so they use that to get the structure, and that is what AlphaFold is doing here. In the physical forces here are things like the van der Waals and the protein, or the potentials between the atoms. And so this is just a little more in depth of what the gradient descent is doing here with the different features. To get in, so this is to get the most likely conformation between protein models. So a full algorithmic design, as we can see here, we start with sort of the sequence and the multiple sequence alignment features that goes into sort of the deep neural network. We get distance and torsion distribution, as that has a large effect on the sine phi interaction between the atoms. That goes into a gradient descent based on specific physical features that proteins can have, which then gives an output. This is sort of the basic model that they have for AlphaFold 1. Is there any questions here before we jump into AlphaFold 2, which is actually a little simpler to explain since it's broken up better into parts? Great. I'll take that silence as a confirmation.
I had one
Oh, go
question.
ahead.
They used
Yeah.
this first deep neural network is the one that they reused, right? From I think it was their which one was it? I think they reused it from another project.
Yes, from my understanding,
Retrained.
they at least started the weights, or they initialized it from. It was I think it was AlphaGo.
I think so too. I think so too. Yeah.
But yes, they did have starting weights from a different project. You're right. With AlphaFold 2, they actually decided not to keep the same weights and started more of they call it a black hole initialization. But yes.
Right. I thought that was pretty funny because apparently they were like, yeah, let's just use the AlphaGo network and just like plug it in.
Yeah. And they got decent results. I think they were up to 67% on the scoring mechanism that they had for this contest with the CASP 13.
13, yeah.
Mm-hmm. But with the AlphaFold 2 architecture, it's three main stages. They have the pre-processing stage, which is mostly the left before the recycling times, the MSA pair representation, and the Evo former, which is, as we will see, a play on words of evolving transformer, and then the structure block at the end, which is more of a residual block for, or is more of a RNN type thing. Great. One thing I do want to note, between AlphaFold 1 and AlphaFold 2, there was actually great debate on whether or not this is important for the field or not. And so many labs were trying to replicate what was happening, which actually made DeepMind postponed publishing this paper for around a year because they wanted to make sure that they weren't scooped by other labs, especially the Baker lab at UW, trying to achieve the same performance as AlphaFold. And so they were really extremely worried about getting scooped, which is fun to think about. So with the pre-processing stage, it is used to take input sequence and query additional information from external databases. This is the things, as we know, such as the protein data bank. And then these are mapped to two different sort of weighted pairings. We have the multi-sequence alignment, the MSA, and then the pair representation, which then get redefined by the Evo former, as we'll talk about in a couple of slides from now. And then finally, the structure module transforms the abstract representations from the Evo former into concrete three-dimensional coordinates for the protein geometry. Looking more into the pre-processing pipeline, we have here our different target features, the MSA features. There's a bunch of concatenations within this block to get to the pair representation and the MSA based on looking up data from known databases. They also try to derive and repeat the process several times with noise to try to get different MSA matchings or pair representations that potentially don't exist. And they do this by running this process several times, up to, I think, 7 to 50, depending on the protein. Oops, sorry about that. And then they average out these MSA representations and pair representations before concatenating them into the Evo transform. And they found success in repeating the database search to concatenate these MSA pairings and pair representations. Looking at the Evo former, the second part of this algorithm, this is 48 blocks within this module, each having individual parameters, so it's not a recurrent network like the final structural model is. There is a stream of MSA representations and then a pair representation. The MSA representation is attempting to do both row-wise gated self-attention and column-wise self-gated attention, and this is to learn the spatial and evolutionary pairing of these represented structures. Going a little further into that, they call them axial attention blocks, where the row-wise and column-wise, or sorry, where row-wise aligns each of the aligned sequences separately, looking for similarities, where the column-wise reasons between different residuals with different sequences of multi-sequence alignment. So row-wise is essentially trying to look at each sequence, where column-wise is looking at interactions between each sequence. This operation is performed on all, and so after that, then you go into sort of this outer product back into the pair representation of this algorithm, where you're doing triangle updates throughout the sequence, and that looks more like this. And so here we have a example of a histogram where the I-J pairing is the 2D mapping of the residual between residual I and residual J. And so here, this is just a symmetric matrix on the identity, or on the column, or sorry, on the diagonal. And so if we sort of know that there are distances between IJ and IK that are not physically represented, so when you're making a triangle, is the hypotenuse too long for the two other sides? Does that make sense? They do these calculations to make sure that any kind of interaction between the known points are actually physically plausible. And so they do that here by triangular self-attention with the starting node. And this is some very thick math in going into each of these specific triangle modules within the supplementary section. But essentially what they're trying to do with this feature design is to make sure that it is physically, physically plausible for the interaction between residuals.
And they corrected, right?
Yes, and they corrected, yes. Sorry, that was just a mouthful, and I wanted to make sure and stop here if anyone had questions. So essentially what this Evo former is trying to do is both take the spatial representation of the protein structure and the evolutionary conservation between the pair representations of the residues to make sure that they all conform to the current protein in question. And so once they get that, they are outputting abstract representations of both the MSA representation and the pair representation, which the final component of AlphaFold will take in these inputs and output a concrete 3D structure. So this structural layer, each layer of the MSA representation feeds into an 8 network layers that all share the same weights. So essentially this last part of the algorithm is a recurrent neural network that is iteratively updating the structure of the protein. These fundamental constraints can disobey the laws of physics. They have an example up here with E, where they don't necessarily align with physics, but they find that using this information trained on this network, it actually helps the accuracy on the data that they're using. So the author, go ahead.
Sorry, so can you repeat that? So they are allowing the law of physics to be broken because they want a metric to be higher?
Yes, they are. That is a thing that they actually did.
Okay.
So with that being said, most of these structures, so we have to think that our information about biology and physics is incomplete, and so they may actually be real structures, but it's also sort of a black box representation, and I wouldn't take that to heart. What they also do, and I think I have it in a future slide, is they augment their training set as well, because the data that is currently trained on is only biologically known proteins, and they've actually found about 150% worth of the data, more protein structures that aren't necessarily accounted for, and they use that data as well, which sounds like overfitting in my mind to better perform on this data set. So yes, they are disobeying the laws of physics with this part of the algorithm. Does that answer your question, Ashkan?
Yeah, it's answered more than enough. Thank you.
Yeah, no problem. And so as they go through this, how they do the initialization of this structure is they start with what they call black hole initialization, which means that the structure module will initiate each of the residues at the exact same point of origin, and then update the model accordingly. So everything starts at the same point with the same torsion, the same angles. They use quatrions, which is a way of measuring a space with 4 angles rather than 9 coordinates in Cartesian. And so as they are learning each position of the residues, they're only learning 4-dimensional vectors instead of 9-dimensional vectors. And this is also good when training this model due to the smoothness of angles. So instead of bouncing around with different coordinates, since they are in a quatrion space, all of the angle changes will be smoother, which helps this final gradient descent to learn exactly where those updates should be. Once initialized, there's 2 different stages that goes through this part of the algorithm, the IPA module or the iterative point attention model, which updates the sequence representation without changes the structure of the sequence. In the supplementary details, I think there's about 10 pages just on this module. It's a lot of tricks to try to keep it point invariant while using some different attention heads. I think it's 8 different attention heads in this one, but if you are interested in this, it's pretty thick in the supplementary. But then this goes into an equivariant transformer, which basically updates the sequence representation and keeps the activation weights invariant rotations and translations, much like many of the computer vision problems that we've been seeing in recent years and months. This way, it keeps the protein structure invariant to rotations and translations, which is great. I am blasting through, sorry about that. Are there questions about this so far? I know this is a very thick paper.
I have questions if no one else is ready. So follow up to Ashkan's question. The whole thing outputs proteins which are physically infeasible, or inside they allow, while they're optimizing, for proteins to take non-realistic, like whatever, non-physical positions, but when they output, they're back to the physics.
Yes, and they do that specifically by adding a final shallow layer here, or a shallow ResNet layer to predict the torsion angles between each residue to refine the structure to keep anatomical physical properties.
So the actual solutions are always physical, right?
Yes.
Okay, so it's just to bypass, it feels like it's to bypass local minima. So they're not trying to simulate the full scale of actual folding, but they're still producing what's possible.
Yes.
Okay, thank you.
Yeah, absolutely. Other questions before I move on? This is essentially the whole part of this algorithm.
Can you explain the, I don't know what it's called again, quaternions.
Yeah, I didn't put up a good visual, but essentially it is a 4 vector angle representation of a point in space. Instead of when you have 3D space, you'll have 9 points that you'll need to get one specific point within the 3D space. You only have 4 angles to keep a part of when trying to figure out that space. Does that make sense?
It's a generalization of complex numbers.
Yes.
And
yes.
like 4D or like n-dimensional.
Yeah, and it's probably the only possible generalization of complex numbers. But Hamilton didn't know about that, so he thought there will be continuum, and he's a physicist, Hamiltonian, and everything, you know, of fame. But he considered this his largest invention, but it didn't take off as much as it has potential. For Clifford has done something with it as well, but.
So
Or no, it's for 3 dimensions, by the way, I think, not for n dimensions. I think it's just for 3 dimensions.
It
Yes,
is.
yes, it cannot be generalized for n dimensions.
Yeah,
This
exactly.
is clearly spatial, but it's generalization of complex numbers still.
Mhm.
Yeah,
Wait,
exactly.
for
But I
like
think Renault said n dimensional.
yeah, I said n, but like, for example, if you have like 2 dimensions, and if I have to generalize it in terms of this complex numbers, I basically go from 0 to 180 degrees, right? Or 360 degrees also. But what my confusion here is, how do you, what is that 4 angles that you're talking about? So are you talking about like 4 angles that
So sorry, it's 3 angles and a length. I should be clear about that. So it's each dimension that you have. So the X dimension, Y dimension, Z dimension, and then a length.
right? Okay, that makes more sense.
I think it's like in regular complex numbers, you have ij, and in 3 dimensions, it's ijk.
Mhm.
And if you just add ij, k, and d, you would have like 1 more dimensions in the complex space. So as Sergey said, this is probably the only one that has a valid complex multiplication you can define over that field. So above that, it doesn't work anymore, like more than 4 dimensions.
Probably there are no higher order generalization.
And this is just to use, I mean, just to take it out of the map or the minimum, or that optimization thing.
Although I may be wrong, but that's my belief.
I think it's just a really common way also in game design, for example, to represent 3 objects, because you only have 4 things that you need to predict now instead of like 6 or 7.
9.
Like,
Yeah. And it also like helps with rotations and stuff. Yeah.
Exactly. Because normally what you would have to do is have X, Y, Z, and then rotation along X, rotation along Y, rotation along Z. So that's 6. But you can do it in 4 here, which is just more efficient, basically. And I think it's also more closer to each other. Like points that are closer to each other actually mean they're closer to each other in distance.
Yes, and that is actually one of the big bonuses of using quaternions, is because of the smoothness within the processes itself. And
So it does help
I'm-
optimization in that way.
This is the first time I'm actually hearing about this being used for an optimization theory. Is there any, I mean, do you know if this kind of theory has been used in any other algorithm
There is a book in my office. I'll give it to you after.
which uses this?
Yeah, honestly,
All about
first time
that.
I'm hearing about it too. But it's interesting because it is a 3-dimensional problem. And so thinking about using the different coordinates just makes sense. It's the difference between like using RGB and like HR or HSV.
Yeah, HSV, for example, would be better for optimization because points are closer to each other because it's a circle. And that's also with quaternions.
Quaternions, I'm sure, were used in Quake 1 engine or something like that.
They're used in all modern 3D generations. They're used, I think, in all modern 3D physical generation things.
Cool. But you all have been looking at still images so far. Let's see if my videos will play. Okay, so the second one plays. But this is in real time, or not in real time, but this is just the structure it's predicting at each of the blocks for the different iterations it's going through. And so it has 48 blocks that it's going to, and then I think this one is going to 7 or 4 blocks, it looks like. But yeah, this is it in practice. One big thing that we should talk about is, well, how is the algorithm trained? What is the loss components? And they came up with sort of 4 different components that need to be adhered to in their loss function. With a major one is frame alignment, or frame aligned point error. And so the second half here has the computed frame aligned point error. What it's trying to do is without getting too in the weeds, it wants to conserve substructures rather than atom placement. And so it's weighting heavier on substructures being conserved rather than the change of individual atoms, as we know that evolutionarily, the structure should be conserved before atom placement and genetic altering of those different placements there.
How do they determine how do they determine the substructures that they want to conserve? Are they just like common ones, or?
Yeah, so what I believe is that what I think that is going on is different residues have very specific types of structures that they already have within the database that they can glean from. And so taking that into account, they have they have two different regimens. They have a training and then a fine-tuning regimen. And so here at the top, this is the loss function they use for this algorithm. For those who maybe can't read, the first one is the FAPE. Second one is an auxiliary function, which has random massing within the algorithm itself for the algorithm to try to relearn different parts of missing parts of the structure. It has an MSA one where it has the distance measure with cross entropy attached to it. And then it has a confidence level where it's trying to see how close it can get to the anatomical structure as well. When they're fine tuning, they actually use these other two problems, or the other two pieces of information, where they're recycling data and then using non-biological proven protein structures as well. So data outside of their distribution that they have generated themselves. And it looks like it is actually helping with the algorithm and the fine tuning. In these two, and then the violation loss defined is one specific to physical interactions of atoms, which is why they wanted to use that as a fine-tuning part of their loss function rather than just training in general. So
I have a question about this.
Yes.
Weight coefficient for the losses, because like with multi-loss training is always difficult, right? Like one can dominate others. How did they set them up? Why 0.5, like, and why different for fine-tuning? Oh no, fine-tuning, they just added more losses.
Yeah, it was a brief section in their discussion, but from what I could glean is the first two loss functions, the FAPE and then the auxiliary loss, it's specifically at 0.5 because they want that to learn faster or than the others, but it also looks like the MSA loss is at 2.0. Sergey, to answer your question, I probably have to go back and look at the details.
That's fine. That's fine. Just briefly, yeah. If you remember. But I have another question. Do they ever talk about how they train it? Is it by curriculum? Because I'm always finding curriculum helps, or they just like, okay, set everything up and run the same thing, or they have like a simpler problem or different problem first and then increase the difficulty of the problem. Anything like that?
Yes, so they actually did a bunch of different regimens. So here to the left, they actually showed with self-distillation training, that's with adding the non-biologically relevant proven protein structures. They have their baseline, no templates. They have no raw MSA or any kind of templating that you're talking about. The direct projection of the point invariant algorithm, they don't use any of that. They have this thing called recycling, which is using data that they have created to just feed it back through the network to update the weights a little further. I'm not quite sure how many times they use the recycling method within that. But what they've showed is that self-distillation training, or basically running the whole algorithm through finding these new proteins, adding them to their sort of data bank for the initial pre-processing, has actually helped the most in terms of getting to good projection of protein structure. So that is what the left graph is showing. I don't know if that answers your question, Sergey.
No, this is more like ablation study, but I was thinking maybe they kind of go through the stages of training on different problems of different difficulties, but it looks like there's only 2 stages, pre-training and fine-tuning, right?
Yes, yeah. So to answer that question, I think you're right there. But yes, this is the ablation study, which they show is smoothly going to around the best prediction that they can without improving.
Yeah,
great. Yeah, of course. So we've gotten to the point where we can ask, is protein folding solved? Bottom line, absolutely not. But it is a monumental and a great start, especially getting over 50% prediction. So it's slightly better than a coin flip. I believe they got to around 88% accuracy in their measured procedure. The prediction, though, is not always great, especially for non-biologically relevant protein structures that are needed in sort of pharmacology or pharmaceuticals or any kind of like biodegradable substances, like Eloy said about like the plastic being dissolved. Those are very specific and have different interactions that are not captured by this database distribution. And then there's even the part of many interactions between complex protein structures. Like you see here, there are 3 proteins. They actually change the folding that they have to interact with each other to best utilize their structures in a machine-like organism. So a lot of times we have the different ion channels that you can think about in a neuron, and those are all built by proteins. And so those have different structures than the individual proteins that are with those ion structures. And then can this generalize? Not yet. They think a lot of the relevant protein structures are not captured in the database that they use to train it on. So there's just further work that needs to be done with this. But great monumental feat. And I know I've been talking for about 50 minutes, and that is all that I have for this. There's about, I think, 10 other papers on AlphaFold since this was published last November. For different people trying to parse this out. They also have a collab book or collab notebook that's ready to go and actually to use AlphaFold. So if you want to do that live, you can just do that even after the meeting. So everything is open source, everything is online. But yeah, we have about 10 minutes if anybody has questions about this.
I think one thing that's nice is that they add a confidence number with each prediction. So they've like created an open data set of all known proteins.
Yes.
And their 3D structures, and then
so it's not only
that the algorithm has.
Yeah, it's not only the competence of the structure itself, but it's individual parts of the structure. So it's every residue that's a part of the structure has a confidence interval of being correct given the sequence.
Yeah, I just shared that link in the reading group. So maybe if you want to like open it. So the database it has like the structure of each protein along with 3D view and model prediction. Yeah, so if you guys just click like if you scroll up, yeah, these in the example part. No, just scroll down just a bit. You'll see that in the example like free fatty near the search engine.
Sorry, I'm okay. So
Yeah,
right under.
Yeah.
I see. There we go. Okay.
Yeah, so if you just click on any one of them and just scroll down once. Yeah, so here's a. So they're given the model, the confidence, the 3D structure as well as the view.
Nice. Yeah, so it's the heat map here is what we're looking at. That's great.
Oh, I do have like I don't know if they've implemented this or not right now, but do they talk about combining 2 proteins together, like calculating high infinity RNA?
Yeah, this was in their discussion section since this is still a very open problem. They don't know how 2 proteins are going to interact given 2 sequences of amino acid chains. So that is still a very open problem, and I think exponentially more complex than this problem.
Because like I have read few papers where they solve this problem of RNA affinity using a graph neural network, but I'm not sure if that's been done using this AlphaFold. It would be really interesting if they do it using this.
Yeah, I guess being a little naive to the chemistry field, I don't know how much data is accessible by protein interactions. I'm sure there's plenty out there, but I know the real big crux of this problem specifically is because of the last 50 years, they've generated upwards of like 3,000 biologically relevant protein structures with all the metadata about the molecular structure, the atomic structure, all the interactions between residues. And so because of that information, they were able to train on this. And from my knowledge, they did a 70-30 train test split.
Okay.
Yeah. Oh, cool.
Yeah, I think mostly what's nice is that that this can help people that, especially in fields where there's not a lot of money, to help at least have some idea of what structure may look like. I mean, obviously, like you said, the generalization is still like under question because we just don't know for sure.
Yeah.
But there are, for example, I think there's an institute with like long, like sort of forgotten diseases, especially in countries that are have less money and resources for research. And in those countries, this can be helpful for those specific diseases, maybe, because they at least have some idea of what the protein will look like, and that will help them select which protein they will look into further with like crystallized, I think it was X-ray, right, or something. At
Yeah,
least
they
do more research on.
Oh yeah, and they did actually already publish a paper since last November on a structure using AlphaFold that is now novel and new to the field. So
Yeah, exactly.
this already has applications. And I think they have 8 patents on this technology already. They went a little crazy. In the paper, they claimed, I think, 4 patents, but I looked it up and it was like now up to 8 patents and a spin-off company. But the best thing I think about this, and especially when looking at sort of the descent of this paper, is that there are other groups trying to replicate and stay up to pace with DeepMind, so it doesn't become monopolistic within this research field. So they don't necessarily own all of protein folding technology for the next 10 years.
I think UW already published like a paper that was more efficient and better, right? Like in terms
Yes,
of their
yeah,
metric.
the Baker Lab is very huge in that, and they're so open source.
Yeah, so I think that's good at least. But I think I would be interested in, are they going to do a post hoc analysis on why this works on this metric?
I'm sure it'll come out in NeurIPS next year. Or just another Nature Communications with David Silver's name on it.
Yeah, like trying to understand like why it seems to do well at predicting, like to understand the process of it sort of folding proteins.
Well, I guess that's why they use, or at least they deemed it the Evo transformer or the evolutionary transformer, because they have the assumption that structure is conserved over generations of evolution. And so they're able to match the same, or that multiple sequence alignment. They're able to match the amino acid sequences to evolutionary lines and then capture that evolution conservation in the Evo transformer. And that's their big like claim to fame, I think, with the AlphaFold 2 algorithm. Does that make sense?
Yeah, I think it is also nice because it builds very, very heavily, extremely heavily actually on just prior knowledge in the field, I feel like.
Yes, it is. This is definitely so much prior knowledge to the field, and it would not be where it is without the 50 years of work towards this.
Which is also cool to see because AlphaFold 1, I feel like at least to some extent, is more of like a plugin We take this ResNet and we try it on, but then I think it's encouraging to see that things can be more efficient and more performant, at least on this one metric, when you do use the, I guess, like hundreds of years of research that we've been doing in the field.
Brings me to something that we started this meeting with, was talking about fusion or nuclear fusion and sort of different applications to that. I'd be curious sort of if they have an Evo transformer for fusion or what kind of information is out there about fusion data. Does that make sense?
Yeah. No, I think that's really important, because it will also help, personally, I think, make this more interpretable. Like, because there is some sense of like the MSA features, right? We do kind of know what they are. Like it's not just like a complete black box.
Yes.
I think that's also really nice about this.
Yeah, very nice. Any other comments, questions, concerns about this work? But I see that like a tendency of sort of quote unquote correct usage of this new tool, deep learning, right? Where we have physical models, so we know they're correct and we know they're fast. We use them where our physical models are unable to perform because of uncertainty or because of other reasons. We try to replace them with machine learning, basically learnable models. And in concert, they produce much more interesting results. That feels like the pattern.
Yeah, but I also think it's very important, like for a lot of these things that work better, it seems like there is, because I had imagined that their idea of MSA, like this evolutionary thing, I imagine that that's been done before for the CASP.
Yes. Oh yeah, absolutely. And a lot of it seems to me, and especially with CASP, it feels like it is at least overtraining to, overtraining to the data set needed, overfitting, that's what I was thinking of, which really struggles with generalization in novel cases that might be more beneficial to clinical trials.
Yeah, so I think what I was trying to say more is like, I think it's cool that they open up a few of the extra parameters in these models basically, and then try to learn them using gradient descent, because it seems like in essence, it is very much still kind of that physical model that other people are using, but they make it a little bit more kind of like, they open it up basically a little bit more, and they add, basically they try to train it with gradient descent and some other modules. A lot of different modules.
Yeah, this may be my naivete in sort of the current workings of the field, but they did not use ReLU for their activation functions. They used ELU or ELUs or exponential leaky units or linear units. I'm just curious, is that a new trend that's going around? I started seeing that.
Yeah, it's very good at seeing. It's very good at self-normalizing.
Okay.
That's basically the idea. They're used a lot in VAEs because VAEs are very unstable. And I imagine that's also the reason why they use it here, because if you have like it's a very long model. So if you have any instabilities early on, that'll just ravage through your whole network. And I think that's why they use them.
I see. So then why would they use leaky ReLU?
Because ELUs are basically a nice sort of like smooth way. So you don't have this, the gradient is just a little bit more cut off in leaky ReLUs. Okay.
Yeah. Okay. That makes
sense.
If
you look at transformer training, the Gaussian linear unit, GLU or whatever they call it, they're also dominating now, and people are trying to port them back into CNNs and such. But,
interesting.
The idea is that it normalizes the activations to a normal distribution for GELU at least, and then for ELU it's kind of similar, but not specifically a normal distribution. Sorry.
I think we're ignoring something. Those activation units were known for quite some time, and I think even in our conversation right now, we're pretending as if those are independent parameters. We're trying to rationalize why just this thing works by itself. But I think we've discovered something else that now is beneficial suddenly those previously existed things are working well together with it. And by focusing just on kind of non-linearity, we're missing the larger picture. Something like it's multiple pieces acting together and working better in this combination rather than with reuse. That's just what I suspect.
Oh no, for sure. But I think TJ's question was more like, hey, I just didn't know this one before, or I haven't seen it often. What is it?
Yeah.
I don't think anyone is implying that it's because of the ELU units that this is working better or worse or whatever.
Yeah,
But I think it makes sense to use I mean a better activation function for this kind of network, because I mean even if you use exponential leak period, or even in your case if you use just GLU thing, so the amount of data that we have and the amount of activation that we need, it makes sense. But if I for example, if I would have used normal ReLU, I wouldn't have gotten a better performance in comparison to what I'm getting right now, because just because of the performance of the activation function.
Mhm.
So
Yeah.
I think the what Bruno says, activation function doesn't matter. Move on. No, I'm saying it doesn't matter, but for a specific problem, we have to choose it. We can't just blindly choose any activation function, is what I'm saying. Yeah, what I'm finding when in my two examples that I also often forget, there is some certain hysteresis in the system when I'm experimenting. I'm not doing grid search for all possible parameters, and I have arrived suddenly at this architecture and set of parameters that works. I don't remember how I came there, right? And it works now, so I'm moving on with what works. Well, anyways, I think we're out of time. Just 2 minutes out of time. Thank you, TJ, and thanks a lot. That was very nice. Thanks everyone for joining. Yes, see you next time. Schedule is online.
Thanks, everyone.